All right, so as I was mentioning, we have these genes, right? For super taster, non-taster, taster. And so that's the first thing that we're going to do is find out if you are a taster or non-taster. Now, I want to try something. In the bags that you got, there is a strip of paper. Don't get it out yet. That strip of paper is filter paper that I soaked the probe on, so it has the probe on it. But I also found, because these didn't used to be available, some commercially available probe strips. And so what I want to do is see if there's a difference between the ones I made and these commercially ones, or if I can use these and not have to make my own. So you guys are going to help me with that experiment. So I will hand those out in just a minute. Before I do that, thank you. The next part, after we do the probe tasting, right, then we're going to look at how another psychophysical method, right, about how we rate intensity and whether or not the scale that we use affects those ratings. Okay, so we're going we have different sweet and salt solutions around. There's 20 of them around on the different tables. And what we're going to do after we do the probe is we're going to use what's called a labeled magnitude scale. So if you look at your handout that I gave you, this is a labeled magnitude scale. It's got magnitudes on it. It's labeled with magnitudes, just like it says, right? And to use that, what you do is, so here's your scale. When you taste your probe, if you think, oh, I can kind of taste that, that's moderate, then you would just put like an X or a mark on this scale. So you put a mark like wherever, because if you think it's very strong, you put a mark there. So you're marking across this line. These have tick marks and these don't, that's fine, right? Across there, and it goes from barely detectable, right, all the way to strongest imaginable. Now, strongest imaginable means if you were to put another taste in your mouth, nothing could be stronger than what you're tasting right now. Like that's what it means, strongest imaginable. Can't imagine something being more stronger than that. So after you're done with all your other rankings, you're going to use one of these pieces of overhead sheets that are up here. I have one for everybody. And you're going to lay it on top and match up the labels. And that's going to allow you, it's going to put these numbers over. So if your mark, say you marked it, right, you thought, oh, that was pretty strong, so I'm going to put an X there, right? Then when you lay that on top of it, you're going to say, okay, that is probably a 3.7, right? Right? And so we get a number associated with that. But just looking up here, by show of hands, how many people were in the blue, like moderate and lower? Okay? All right, so you guys are probably my non-tasters, right? Didn't taste like much, right? What about in my orange, sort of in that sort of moderate to very strong in that range? It's like another 4 or 5, okay? And then everybody else was very strong or higher, and so you're probably super tasters, right? So that's probably the way that that's gonna break down. If we did a little bit more extensive testing. And like when you really do this test, if we were doing this for publication as real research, like Linda Bartoshuk does, this is her area of research, then we'd have you sipping solutions of probe at different intensities because that works better than these filters. But this is a quick and easy way to kind of do this, okay? All right, so what's the big deal? So as I mentioned, Linda Bartoshuk, who wrote your chapter, your taste chapter in your textbook, her area of research is in human psychophysics, and in particular, she's looked a lot at probe and correlations of probe with other things. So she looks at really big data sets, like thousands of people, and looks at these relationships. And so one of the things we know is that if I took pictures of your taste buds, my super tasters would have more fungiform papillae than my non-tasters, and the tasters would be somewhere in the middle. And yes, there's individual variants. Sometimes the non-taster has a lot of taste buds. Sometimes the super taster doesn't have very many. But overall, if we look at a big population, there's this positive correlation that says the stronger protein sensitivity is, the more taste buds you have. So the more overall transduction mechanisms you have and a bigger taste signals going to your brain, right? Some other things that Dr. Bartoshuk has looked at are relationships with other things. And so it turns out that sometimes super tasters are more susceptible to some health risks because they tend to shy away from bitter foods that have some positive health benefits, like broccoli and grapefruit juice. And a lot of bitter sort of foods have antioxidant types of protection things. Also, there's a trend for super tasters to sometimes have higher rates of obesity. We think because of sweeter taste sweeter to them, more repetitive, more rewarding, and so that could be something associated with that. Non-tasters also have things associated with them, like there tend to be higher rates of tobacco and alcohol use with non-tasters, and we think probably because the negative oral sort of perceptions of alcohol and tobacco are lessened for them and not as aversive, and maybe more aversive for super tasters, and they tend to shy away from using that. Right? All right, so, and on the Moodle page, all right, so there are two reference articles on here from Linda Bartoshuk for you to use in your introduction. And I wanted to show you the first one has an example. So the first one talks about the differences in scaling and talks about, remember Fetchner and Stevens that we talked about very first day? Talks about differences between using a 10-point ordinal scale and using the Green's labeled magnitude scale that we're using. And so that's the first experiment that we're about to do. And I'll go into that in a little bit more detail. But I wanted to show you this graph because this is the sort of thing, the sort of stuff, research that Linda would do to show you that if we look at a whole lot of people, right, that they tend, as the bitterness of prot goes up, so our super tasters over here, non-tasters on this side, they also rate the sweetness of, in this case, a butterscotch candy. Non-tasters tend to rate it as less sweet. Super tasters tend to rate it more sweet. Now, this correlation is 0.3. And so you see like it's a slight incline, positive correlation. So these, and you can see like how many data points are on here. And clearly, some super tasters that rate prot very intense rate it very low in sweetness. Some non-tasters rate it very sweet. So there's a lot of individual variability. It's not like these genes govern your taste system, right? But, and all of the data pretty much with super taster stuff kind of looks as messy as this is, right? So when we talk about these genes, don't be like, oh, I'm a super taster. I'm going to get cancer because I don't eat broccoli. It doesn't work that way, okay? All right. All right. The other paper I'll talk about, I can just go ahead and mention, has to do with the last experiment that we're going to do with miracle fruit and Genema sylvestre. And so I'll talk about that when we get to the end of the class. All right. Back to work. All right. So four parts of the class, of the thing. We actually did the first part. So the first part is measuring your fungiform papilla, which we're not doing, and the prot sensitivity. The next part is going to be going around and tasting each of these stimuli that are around the room and rating them on the label magnitude scale. That's stimuli 1 through 10. And then if you flip over past that, you'll see here for stimuli 11 through 20, you simply write an intensity from 0 to 10 for each of those. Okay? These are, all of these solutions are either sugar water or salt water. They're fine for you to consume. You can also take a cup around with you and spit it out. If you don't want to consume it, you just need enough to taste it, to rate the intensity, the perception. So this is where you're going to put in your numeric rankings, right? 1 through 20. And at the very bottom, down here, the last one is where you're going to put the probe rating. Okay. The third experiment we're going to do, so after we're done with that, we'll clean everything up. When you, there's a stack of cups for each sample, so get a new cup for each sample. In between sampling, have a drink of your water, because we don't want adaptation. Like you drink a really sweet solution and then the next one is a real weak sweet solution. If you still have that high sweet solution activating your mouth, then it's going to interfere with the lower one, right? So take a little water in between. There's trash cans around to throw your cups away. Just stack them up as you go and just throw them in the trash can when you get near a trash can, okay? The second one, you guys are going to work in pairs, and I want to keep like at the ends of the tables distanced, okay? And so Aaron, I'll have you pair up maybe with Anita, and you guys can do like back and forth there, something like that. So what you're going to do is each of you is going to be a subject, and each of you is going to be an experimenter. And we are going to look at the interaction between smell, taste, and flavor. And so when you are, so what you want to do is, like, so if I'm the experimenter, this is my data pack, but I'm going to put my subject's initials, and I'm going to put their data under their initials. That's why I had everybody put their initials in there, right? So if I'm working with Lexi, and I'm recording her data in my sheet, when I go to type in the stuff, I'm going to put my LMS and ordinal sweet and salt scores under my initials, and then I'm going to find Lexi's initials and put them under her, and put the flavor stuff and taste stuff under her name. Cool? So we're not switching packets and all that kind of stuff. Okay. So when you're the subject, you have two Ziplocs of jelly beans with 6 colors each. And so what you're going to do is you're going to put the blindfold on for both parts. In the first part, you're going to have smell and taste. So you're just going to pick a jelly bean out and you're going to show it to your experimenter. So if Lexi picks out a jelly bean and it's the red one, she shows it to me. Then I'm going to the red column here, and she's going to taste it and tell me 2 things. She's going to give me an intensity rating, 0 through 10, like how intense is the flavor. And she's going to guess at what color the jelly bean was, right? So if she guesses green, I'm going to write green there, and that's going to be an incorrect answer, right? If she guesses red, then that's a correct answer. So once you're done, you're going to tabulate the percent correct, however many out of 6 they got correct here. And then we'll have an intensity level for each one. Good? For the second part, so what you're going to do, excuse me, is repeat the same procedure with your second Ziploc of jelly beans, except this time you're going to hold your nose shut. Now, you need to hold it shut really tightly, and you can't let go. And I'm not going to lie, it's a little difficult to chew a jelly bean because you have to breathe through your mouth. So anyway, you can do it. We've been doing this for years, okay? But if you let go at all of your nose, immediately, so holding your nose like that keeps the retrograde passageway from being activated and doesn't allow anything to come up through there. Don't have that in here. No, don't. And if you let go at all, immediately you'll get that smell perception. So we're going to redo the guessing the color and the intensity with just taste. We're blocking smell now. And we'll look at the contribution of smell and flavor. Good? Any questions? All right. If you don't want to eat the jelly beans, use one of the sample cups and just spit them out. I don't care. You just have to taste them and guess the color. We need intensity and we need color guess, okay? Cool. All right. And then the last thing that we're going to do is look at the role of transduction mechanisms on our tongue and with some taste modifiers. But I'll talk about that after when we get there. Let's get going on this. Okay, the last part of our lab that we're doing today involves the dulcine berries, which are a plant that's found in Africa. And it's also known as the miracle fruit. And from these berries, there is a chemical called miraculin that you can extract that is a taste modifier. And then there's also Gemina sylvestra tea, which is another taste modifier. So if you remember when we talked about gustatory transduction, taste transduction, that each of the primary tastes have their own transduction mechanisms. So for bitter and sweet, those are lock and key mechanisms, right? For salt and sour, those are ionotropic channels. So sour acids come in and salts come in, right? So what miraculin does is it blocks your sour transduction mechanism. So it's basically like it eliminates the perception of sour to go to your brain. And at the same time, it binds to your sweet metabotropic receptor, right here, that T1R2R3 receptor, in a way that when you put something acidic in your mouth, something sour in your mouth, it stimulates the sweet taste receptor. So sour things become sweet after taking the miraculin. The Gemina sylvestra works in almost the opposite way. It blocks, it sits in the sweet transduction mechanism and doesn't allow it to work. So essentially it eliminates your ability to taste anything sweet. All right? So in this experiment, what we're going to do is flip over to your back page. You have an LMS scale with numbers on it, so we can use the LMS labels, but we're going to write down numbers, right? And you have different food items. I don't have strawberries, I'm sorry. But we have some that are more sour, like lemons and pickles and beets, and some that are more sweet, like Hershey Kisses, maple syrup, grape jelly. And so you should have 3 samples on your plate. Now the grape jelly and the syrup are in a cup, and so you're with a spoon, so you're just going to take a sample of that. You just need to get 3 samples out of it. Again, just enough to be able to rate the intensity. And what I want you to do is first we're going to do regular taste, so you're going to taste each of the things regularly. So for the women, you're going to say how sweet is that on the scale, how sour is the lemon, how bitter, and how salty is the lemon. Right? So maybe lemons don't have any salt, and so that's a 0. I don't know. Could be. So you'll have 4 taste ratings for each of those things. And then, when we get to the miracle berry, inside your Ziploc is a little red, it almost looks like a pill. It's not a pill. It's the miracle berry extract. You are not to swallow it. I mean, you can swallow. It's not going to hurt you. But if you swallow it, it's not going to do any good. So what you need to do is, like you do for your COVID test, you want to get some saliva in your mouth. And then you want to put the miracle berry in there and swish it around on your tongue and kind of rub it and get that little red pill-looking thing to dissolve on your tongue. You're trying to coat your transduction mechanisms on your tongue with that. It takes about 1 minute or 2 minutes for it to kind of naturally dissolve. Coat it all over your tongue, right? And then you're going to retaste your second sample of each of those and re-rate sweet, sour, bitter, and salt. And so for the miracle berry, it should eliminate the sour taste and enhance the sweet taste. When you're done with that, drink some water. You can kind of like scrape your tongue on the top of your teeth there, on your top row of teeth, and kind of get all that miracle berry off of your transduction mechanisms. And you can kind of tell if you've gotten it off if you taste your lemon again and it doesn't taste sweet anymore, because the lemon's going to taste really sweet. And when you're done with that, with the miracle berry one, we have the tea that you got a sample of. And that, you just want to swish it around in your mouth for about a minute and a half. And then you could swallow it or you could spit it out, it's fine. And if you're talking the Miracle Berry and you still have a little piece of it left, you could chew it up and swallow it, it's fine. Everything's safe, right? And then you're going to retest everything now with the Gemina Sylvestre. I would probably just go in this order, except when you get to the Gemina Sylvestre, that's supposed to block sweet taste, so I might go in reverse order because the sweet things are kind of at the bottom of the list. Kind of do those first for that one. I would do that. And so we're going to get those rankings in. There is a separate sheet here, data sheet, whoops, right here for the Miracle Berry and Gemina Sylvestre. These you're going to enter as a row across, okay? And so you have your, just like you have on your page, lemon, sweet, sour, bitter, salt. Then you have Miracle Berry, lemon, sweet, sour, bitter, salt. So just like it goes across that row, oops. So just like those numbers go across this row, that's what you're going to type in. And then the next one will start with the sour cubes candy. Cool? Everybody good?